Pleasure to be here and talking about our engineering research, especially in front of our physics expert audience. I hope I didn't reveal too much of my weakness in physics and theoretical physics, and so I'm just trying to show you what we have been doing for a long time and what we're actually good at, which is creating very unique nanoengineering surfaces to help you study biophysics inside living cells. And as an overview, in this hour, I'm going to primarily focus on two major topics. So what we want to do is to develop a new engineering.
Just the lighting?
Okay.
Okay. And then,
yeah, chop.
Yeah, so that's
this is better. Okay. Yeah. So what we're trying to do in my lab is trying to develop and enable new nanoengineering platform for living cell or live cell studies. Particularly today, I'm going to talk about two topics. One is how do we develop a nanoengineering platform to enhance the sensitivity in living cell molecular imaging, particularly confocal fluorescence imaging, for over 100 or even 1,000 times. How do we improve our sensitivity to that level? And in terms of physics behind this, we're trying to introduce a new mechanism, which is a new optical coupling mechanism. We call Nanoplasmon coupled intracellular optical resonance excitation, or Nanoplasmonic core. Using this mechanism, we demonstrate a very sensitive three-dimensional fluorescence confocal microscopy imaging of living cells, and to do the fluorescent molecular imaging at very early stage of protein expressions. The second topic in my talk now is the cell behavior study and control of flexible polymer nanopillar arrays. I see a lot of our collaborator students. I see Xintang sitting there. Actually, he actually made a lot of contribution to this particular work. So here, this idea is we're trying to create a flexible, a very, very small, tiny nanopillars which can be bended, can be actuated by cell force. And we wanted to use that surface to visualize the cell mechanics and try to use that surface also to differentiate cell behavior depending on the cell types. Okay, so let me go to the first topic, which is molecular imaging. Here, our focus is not really looking at individual molecules, because that's not my research focus. What I'm trying to do is develop new tools to help biologists to study the molecules or single molecules inside living cells better. Okay. So if you look at the cells, this is when I give a lecture to ECE students, I always use this slides because our ECE students are very familiar with the circuit. But here, you actually can see a very intricate circuit inside the cells, which are called signaling pathways. There are a lot of protein enzymes involved here. And the goal of to study a molecule or single molecule in living cells, we're trying to identify individual proteins in those very intricate signaling pathways and trying to visualize them even when they have very small copy numbers. So that's the goal for the molecular imaging in living cells. This is typical what you see using fluorescent confocal imaging for a molecular visualization in living cells. In particular case, this is basically you have a nucleus, probably DNA, stained by one particular fluorescence dye or a fluorescent protein, or you see this green fluorescence, which is probably a green fluorescence fusion actin molecules. So people can use organic dyes, quantum dots, or fluorescent protein now to fluorescently label those particular molecules, allow us to do a confocal fluorescence imaging of cells with molecular specificity. But here, there's a problem with it, because for those of you who actually play with this kind of technology, you know that you have to construct a plasmid with GFP, RFP fusion proteins. You have to transfect the cell with that plasmid and let the cell to express that protein. Okay? But you have to wait for at least 36 to 54 hours to see a very optimum imaging results. You cannot just do imaging right after you do the transfection. The reason why for that is you don't have a high enough sensitivity to see the low number of molecules inside the cells. So what we're trying to do now here is to provide a new tool to replace your Petri dish, to replace your glass slide and allow you to image those molecules even just a few hours after transfection. Okay, which means when they just express a few numbers of molecules in the cells, a protein in the cells, we want to actually give you that sensitivity to see them. All right, so the technique we use is, as I said, is Nanoplasmon coupled intercellular optical resonance excitation, or Nanoplasmon core technology. Let me introduce that. But before that, since we have a keyword Nanoplasmon here, so I will spend probably a few minutes talking about plasma. Okay. Here's what I just said. The Nanoplasmon core, we want to enable a three-dimensional, not just two-dimensional, because most of the cells, especially when you do a large population of cell culture, you have three-dimensional spheroidal structures. So we wanted to give not only just on surface, but three-dimensional fluorescent enhancement for more than 100 folds. Okay. And if before you can only detect, you can detect 100 molecules using the system, then using our nanoengineering surface, possibly you can detect just one molecule. So this is the concept right here. And without changing any current optical imaging systems. So there's definitely two ways you can do better engineering. You can either improve your optical systems, you can add in more cool stuff in there, putting more money in there, and make it more sensitive and with a higher resolution. That's definitely very one good way to do it. But our way is not changing anything in your optics. You can keep using the same confocal system for 10 or 20 years, but what we'll do is do better engineering on the glass slide. We will replace that glass slide or petri dish you use with a new nanoengineering surface, so equivalently give you a higher sensitivity, or maybe even higher spatial resolutions in the future. So what we want to do is that if your old system does not have single molecule detection capability, we want to give you higher sensitivity to enable that sensitivity capabilities. And especially for a fluorescent protein fusion, protein expressions, we want to have a new capability to observe very early stage of protein expression soon after gene transfection. Okay. So let me first talk about nanoplasma. So what is plasma? We know, probably many of you know surface plasma very well. Basically, that is a collective electron movement at the interface of dielectric material and the metal materials. So you would expect you have free electrons in metal, because they're conductors, and they will move in a periodic ways along interface between a dielectric and metals upon the excitation of external electromagnetic waves. But we also know this very famous dispersion relationship, that there's no way you can directly couple your free space propagating electromagnetic wave onto cytoplasma, because their dispersion curve does not have intersection point. That's why people use either prism or high refract index material as an intermediate coupling material, or use nanostructure grading to couple the free space EM wave onto cytoplasma. But you can always circumvent that by doing a nanostructure plasmonics or nanoplasmonics. So here I do a side-by-side comparison between a cytoplasma wave, that propagating wave along the interface, and a localized electron movement bounded within a metallic nanostructures. And so here I will not probably go into detail of each individual equation, but as you can see, for metals, their dielectric constant or permittivity is a frequency-dependent functions. And so if you change your optical frequency or wavelengths, you're going to have different wave patterns on the surface. This is why we can basically confine certain modes, plasma resonance modes, within this small nanoparticle cavities. All right, let me move to the actual engineering side of it. So this is what we make in our lab. Of course, you can always do a simplest way. Just you can purchase a lot of nanoparticles from there's so many companies nowadays make nanoparticles out of silver, gold, or even copper. If you do a dark field scattering imaging of those particles, you will see each of them exhibit a very distinctive color, scattering color, because they have this plasma resonance mode in their cavity. Depending on the actual material and dimension and size of the particle, you're going to have different color emitted from the particle. Of course, that's what chemists do mostly. But as an engineer, what we do is we use nanofabrication technology to make a highly ordered, a highly uniform array of nanoparticle or nanowires, so they can have very defined, very controllable plasmon mode. For example, here what you're looking at is actually scattering image of those array of dots, nano dots made of gold. If you can make a very high uniformity fabrication, you can make really a very uniform color out of the whole array. Okay, and here is the scattering image, dark field scattering image from those nanowires. They actually exhibit different color because they have different width in this order axis. So I believe from here to here you have 20, 30, 40, 50, all the way to 120 nanometer in terms of width. So you can, by changing the actual dimension, you can define what kind of plasmon mode you want to have in that cavity. All right, so this kind of engineering has been demonstrated 10 years ago, even, some pictures from my graduate, when I was grad student, actually, some work I do over there like that. But now we actually make it more fancy, right? We can make large-scale nanofabrication using a new fabrication technology, for example, nanoimprinting technology or nanoreplication technology. So here's one example that is a device made in my lab. We start making large-scale, large area plasmonic nanopore array with very fine features on the sidewall, so that they can exhibit different color depending on you do transmission mode or reflection mode. You see the uniformity is so good. This piece has about it's about 1 centimeter by 1 centimeter large piece. So you can see you only see one particular color either transmitted or reflected from this array. So we can basically make over 1 billion of nano hole as a highly ordered array, highly uniform array on the surface of this flexible membrane. And then the result of that is you can use it for more application rather than just nanoparticle itself. So here I want to demonstrate that using this particular device, we can mimic what we see in a Lycurgus cup, which is an artistic masterpiece created by Asian Romans about 2,000 years ago. What you see here, this cup actually is changing color depending on how do you illuminate light on the surface. The reason why this is a real cup that you can pour champagne or wine into it and drink it. Well, the reason why you see different colors is because on the sidewall, on this glass sidewall of the cup, they blend a lot of nanoparticle in there, probably silver or gold nanoparticle in there. And their techniques are pretty good because, well, imagine 2,000 years ago, they can make a cup exhibit very uniform color. That means they can make nanoparticle in very uniform size, even 2,000 years ago. Okay, and then they blend those nanoparticle with their glass to make this cup. Okay, and if you take this cup inside a room, outside a room, or pour different liquid in there, you will see different color exhibit. Okay, so what we're trying to make is a nano version of that in year 2012. As you can see, if we move light back and forth, you can see the color change from red to green, depending on how you do illumination. All right. So, and what actually good function of that is you can also do very simple molecule sensing on the surface. This is exactly what we want to do also for the cell sensing, which we're doing right now. So today, I only show you the molecule sensing. Here, since the plasma resonance is actually determined by material property, the size, the geometry, as well as the permittivity of the surrounding media. So by changing the actual liquid you drop on the surface of this device, you can basically change the color. So here what we try is just drop different material, air, water, glucose, sodium chloride in different concentrations. There is some organic solvent as well. If you drop those liquid on surface, you can see the color difference just by your naked eyes. You don't need anything else. You don't need a spectrometer. The reason why is they become so sensitive to the refractive index change. So their color shift is all the way from blue to red in some cases. So this is a transmission mode. This is reflection mode, as you can see. All right, so this is the engineering effort we're making in my lab right now, one of it. What we can also demonstrate is you can mix liquid together. You see here we have, I believe, this is air. This is probably sodium chloride. You see some crystal precipitation here. And here's, I guess, it's glucose. This is the oil, because they have slightly different refractive index. But even that, slightly different refractive index, which is 100 or even 1,000th of difference of the refractive index, can give us a very large plasma resonance shift all the way from blue to red. Okay. And we believe we're making the most sensitive plasma resonance sensor in the world so far. And you can actually also demonstrate if you drop liquid ethanol, water, immersion oil on different regions of this Illinois size, you can see the color change differently. Okay. All we need in our lab is just using this $2,000 very shabby microscope. That's what we need to take this image. Okay. All right, here's a demonstration that we can visualize when the liquid starts drying on the surface, or the actual liquid flow on the surface. You can see the color become different. So we have ethanol drying and the toluene drying case. All right, so.
No coffee.
Yeah, well, this is not coffee. This is this is completely transparent liquid. Ethanol. Yeah, but they do have a coffee ring effect. They do have a coffee stain on the surface. Yeah. All right, let me just. Okay, so as an engineering lab, we are actually trying to improve our current manufacturing technology to make a highly uniform, large area device. This is another example showing you that we use a technique called laser interference lithography method to make a 4-inch wafer on the entire surface covered by a structure like this. Okay? This is also a batch fabrication, so you can do the lithography at once. You get all the uniform results on the entire 4-inch wafer surface. All right, here's an example showing we can make a silicon or silicon dioxide nanopillar array with a very high uniformity, and you can then deposit metal. This is probably after metal deposition. You see those metal deposit on the surface. All right, so after talking about the engineering capability we have in our lab, let me really move on to how do we combine this nanoplasmonic substrate with the cell imaging or cell study. So the physics behind this, we're trying to utilize the electromagnetic field enhancement on our substrate. For example, this nanocone array, as I just demonstrated in this slides. We want to build a nanocone array to enhance the local electromagnetic field strength. So we do a lot of theoretical study as well, especially on the numerical simulation using finite element analysis or FTD, finite difference time domain simulations, to predict what's the optimum structure to give us the highest possible electromagnetic field enhancement. Okay. So this is actually example showing you basically we just solving this wave equations using a finite element modeling tool software to help us to predict what will be the hotspot or high EM field enhancement. And here you can see for this structures we have an intensity enhancement electric field for about 100 times or even 200 times. Now comes the question, how do we make them more efficiently and at lower cost? So besides of those very fancy fabrication technique like this method, we also discover a very low cost but a very high efficient way to make silicon nanocone arrays with very high density, very small features. And then we can deposit metal like silver or gold on the surface to create a pseudo-uniform or ununiform, you can say ununiform nanoplasmonic substrate. But in some applications, this is even better than ordered plasmonic array because the following reasons. What we want to have basically is a substrate can give us a EM field enhancement at very large, very broad bandwidth. So if you start with a highly ordered structure like this, if you run a simulation and you find out they have only have high enhancement at some particular wavelengths or frequency, which is not sometimes it's not very convenient for us to do experiments because in that case you really have to find a fluorophore or fluorescent protein has exactly matched resonance frequency with your substrate. So instead of doing this highly uniform way, okay, by the way here you can see if you really control the structure well, you can even get your intensity, electric field intensity enhancement for more than 40,000 times, 40,000 times. But what we want to do is basically have a relatively broadband enhancement to the fluorescence. So we started creating a non-uniform, or what you can, but the size of the cones are quite similar, but the arrangement of them are not in order. Okay. So to make the device, we can also make a large areas. So here is showing a half wafer, and the diameter is about 100 millimeter. So we can make a lot of those cones on the surface of a large area substrate. We can also combine with the photolithography to only make those nanostructure on some defined region. Here you see we can basically make an array, actually small square array of nanostructures. Within the actual square, you can expect you have this kind of nanostructure, but outside the square, you have completely smooth film. Okay. As you can see here, if you shine a laser on the surface, you can see very shiny spot, very shiny scattering spot. And if you shine a laser spot on the other side, well, on the other areas outside the squares, you're going to have much lower scattering intensity. The reason why we call it black silver is because they really, looks like black, even though it has very thick silver layer deposit on the surface. Normally, you would expect, okay, if you deposit 100 nanometer silver film onto a substrate, that would be very shiny mirror. But after you deposit on this high aspect ratio nanocone structure, it still looks like black. So that means most of the lights are either trapped in there, absorbed there, or transmitted through the actual substrate. So we do a reflection spectroscopy measurement to compare our black silver with a very smooth silver film. You can see there's a lot of light is being trapped inside that structures. It's not reflecting out. Okay. That's exactly one benefit we want to have, because we want to have more light to concentrate on the surface where you have your cell imaging happen. Okay. We also did some characterization of our substrate. Here we use both photoluminescence and cathodoluminescence to look at the plasma resonance hotspot on the surface. Particularly for the photoluminescence, you see if we compare our nanostructure or nanoplasmonic substrate with a smooth silver film, you see over a large wavelength range, you have an enhancement of photoluminescence or cathodoluminescence. Okay, and this is a two-dimensional profile of this distribution of the cathodoluminescence. You see, you can find a lot of hot spots in the hotspot here. That's why we also use this substrate for surface applications, surface enhanced Raman scattering applications. Okay, so let's see, how do we enhance fluorescence in two-dimensional and in three-dimensional? Okay, here's a demonstration, the surface enhanced fluorescence. We're not getting beyond the surface yet. It's just on the surface. Directly, while the actual fluorescent molecule are directly spread out on top of the surface. So it has a physical contact with the actual surface. After you spread it out on a device like this, okay, as I said, we have nanostructure inside a square, and we have a smooth film outside the squares. And so we can do a fair comparison on the same substrate, a fair comparison about intensities. You can see a much higher fluorescent intensity inside the squares compared to that outside the squares. So if we plot this intensity profile on this line, you can see the enhancement factor for fluorescence is larger than 40 times. Larger than 40 times. Well, this is actually 40 times with respect to the silver film. If you compare this enhancement to the intensity on the glass slide or petri dish, that enhancement factor is even higher. Okay. All right. Sure.
And so, have you looked at either the excited state lifetime, or have you looked at the photobleaching?
Yes, we did. Right. So here are the slides. The next slide is right then. That's a very good question.
How do you know that absorption of your fluorophore is the same on the surface, not on the modified level?
So how do we know that the number of fluorophore molecules are equal or uniform over the whole entire surface? That's a very excellent question. You can definitely make an argument. You may have accumulation of molecules within this nanostructure because it has sort of a three-dimensional structure over there. That's a very fair argument, and we now have no way to characterize really how many molecules there, but we are actually doing more characterization using other technique. For example, some molecule we can really, or some particles we can track, or we can count using electron microscope. Some fluorescent particles which are bigger that you can visualize them in an electron microscope. We want to see whether you have accumulation of the particle in that space.
So
Okay, good question.
How do you call it surface enhanced fluorescence?
How do we know this is surface enhanced fluorescence?
This can be surface enhanced accumulation of dyes.
Yes.
The factor 40 is how much of that is coming from accumulation versus enhancement of fluorescence.
Yeah, as I said, we're doing the characterization. For the small molecules, we cannot count how many molecules in there eventually. So now we're actually starting, using a small nanoparticle, which is in a fluorescent particle, but we can visualize them in electron microscope. We try to count whether we really have a very significant molecule or particle accumulation in that nanostructures. If you have any other suggestion, how do we characterize it better, then that'd be great. Okay. All right. So the other way we do actually is I probably didn't include data here. We do surface chemistry conjugation of for example, fluorescence labeled DNA on the surface, or fluorescent labeled streptavidin or biotin on the surface, that we can predict what would be the surface density for the molecule, and then we can estimate what would be the actual molecule number on the nanostructure or on the smooth film, and then we can discount that part from it and to get the real enhancement factor readings. Okay. So to respond, pose questions, and we also did the actual lifetime imaging by collaborating with Bob Clegg, using his system to categorize the fluorescence lifetime on the surface, and we find out the lifetime for this particular dye, rhodamine 6G dye, on the petri dish and on these nanostructures become very, very different. And basically, we shorten that lifetime significantly, which potentially can improve the photostability of this dye for many folds. We also did some back of envelope calculation to find out how do we use this lifetime to predict the photostability of the fluorophores. And our general consensus now is with the decrease of lifetime, we have a higher decay rate, and we have a higher quantum efficiency. That's what we measure experimentally. The data is published in APL. But this is not the focus of my talk today. I just want to show you this, so-called surface enhanced fluorescence has been demonstrated by many groups for many years. Okay, that's not a new thing. Okay. So people do use that to do a enhanced fluorescence imaging of cells. In this example, it's not my work, it's from somebody else's work. They pattern this actual surface, one area with gold coating, the other area without gold coating. So they actually see the actual fluorescent intensity from the cell on the gold coating surface are much higher than the non-coated surface. And their enhancement is about a few times, from 4 to 6 folds, because that mirror, that actual simple mirror effect, you just reflect more light out, okay, from it. There's no nanostructure there. There's only smooth film. And so, well, even when you use actual nanostructure there, you probably get a little more enhancement for cell imaging, but still, you have a limitation there to see the enhancement only on the surface, because we know this is a very, this is pretty much a near field effect that you have an enhancement. You have your electrical EM field enhancement basically focusing on the surface. If you move away from the surface, then your enhancement factor drops significantly. It drops exponentially. But here we want to propose a hypothesis here that we can do a better three-dimensional imaging, particularly for spheroidal cells. Of course, when a cell really spread out like a very flat sheet, this will not work. In that case, you're going to use a surface-enhanced fluorescence. But this is our hypothesis based on our observations. In this case, if we use a spheroidal cells and let them sit on top of a nanoplasmonic substrate, and we're thinking, can we couple the cytoplasm wave into the whispering gathering mode inside the cell cavity? If we envision cell as a perfect spheroidal or spherical microcavity, can we excite this whispering gathering mode, the resonance mode, inside the cells? If we can do that, then we can concentrate our light field within the cell, confine the field within the cell to improve the equivalent light intensity inside the cell to excite this light, the molecule, especially in the cell membrane, with a much higher intensity. So that's our basic ideas. So here we did some simulation to demonstrate theoretically, yes, you can couple the free space propagating EM wave into a whispering gathering mode cavity by with the help from a nanoplasmonic structures. We also published a theoretical study on Plasmonic journal. Yes.
How do you couple the light in?
Yeah, so the light can be directly shined onto these plasmonic structures. You can do free space illuminations. You don't need any optics. This is what's shown in the at least in the simulation. Okay. So here we launch a plane wave from this side, and this way it's propagating from left to the right. And here we have, this may be too small, but I can tell you this is basically it is an array of a few nanoparticles connect to each other. And then with the coupling of this, with the help of these nanostructures, we do see a light waves couple inside this spherical cavity. If you remove this, it's not going to happen. Most people excite whispering gathering mode in a spherical cavity by using a taper fiber. They use optical fiber. They actually put the fiber close to the spherical cavity to let the evanescent field leak into this spherical cavity to excite that whispering gathering mode. But in this case, we don't do any fiber. We just basically rely on this nanoplasmonic substrate, use that as a mediator to couple this free space optic wave into this confined optic mode within the cavity. Okay. All right. So you may argue that the cell is definitely not as perfect sphere, right? So we have we also did simulation for a unperfect, which is either spheroidal shape of cells or ellipsoidal shape of the cells. Also see that even though we may not have a perfect whispering gathering resonance mode, but we do have more light coupled into the cell, especially in the cell membrane. Okay. All right. So we also did theoretical studies to show that this phenomenon actually is universal. Even when you just have a very smooth silver gold film as long as you have a matched mode, which means if your plasma resonance mode match with the whispering gathering mode, you can even couple a light into the spherical cavity by using a smooth gold or silver film. But in this case, definitely, you have to work in the UV optical wavelength range. Okay.
Over what are these modes to the shape of the sphere?
Yes, yes, of course, of course. This is actually, this simulation is based on a perfect sphere. But for cell, unfortunately, they're not perfect. So you don't have this high efficiency in terms of coupling. But we do observe in our experiments, we do observe three-dimensional enhancement of the fluorescent intensity, especially on a cell membrane. So we try to come up with a hypothesis to explain it. It could be also due to a multiple scattering on the surface, because we are creating a highly scattering surface. It could be due to highly multi-scattering. And also you have a cell membrane to help to confine that, to help to reflect that light. So you have equivalently higher light excitation fields in unit time. All right, so I'm probably going to skip this. You can read the paper. This is not today's focus. So what I want to actually, time passed really fast. All right, to show that is, we want to use this to help us do a better cell imaging. So what we do is we culture the cell. Normally, you actually culture the cells on Petri dish or on glass slide. So we culture cell on this particular substrate. The cell we use is CHO cell, Chinese hamster ovarian cells, okay, CHO cells. And so this is a bright field image we took the first day in our culture. This is from the second day of a culture. You see the cell does proliferate, the cell does not die. They're actually proliferating, the number increase. And also, what we want to have some cells travel, migrate from the smooth surface into this nanostructure surface within the squares. We do see some cells get in there. That's why we actually can find some cells sitting on the nanostructures. All right, so this is SEM we took for the cell start to grow on the actual nano surface as well as smooth silver surface. Then we did confocal imaging of those cells. We stain all the cells. You see there's a lot of cell on the surface, right? A lot of cell on the surface here. Some cells on the smooth surface, some cells are in the on the nanostructures. Now we stain them the same way. We actually treat them the same way. We label all the cells in the same way. We use a red dye, which is Invitrogen FM1-43FX for cell membrane, which is I believe is a dye particularly labeling the bilipid layer in the cell membrane. And we use Horus dye to label the nuclei. So you can see that the actual nucleus and the membrane of the cells. And then we compared it. We do three-dimensional sectioning of the cells. You see these cells are sitting on top of this nanostructure surface. The other cells are sitting on the smooth surface. You can see a very obvious difference between them. What difference? You can see the nuclei from all the cells very, very clearly. Right? Then you can count, we have 1, 2, 3, 4, 5, 6, 7, 8, maybe 8 cells here. But you only see the cell membrane clearly for this particular one. Okay? For this particular one. This is a very fair comparison that we actually, well, this is not labeling any protein, just labeling bilipid layers. We assume that even when the cell is sitting on the top, the cell membrane itself, the actual bilipid layer should remain almost the same compared to other cells. So we actually are seeing the enhancement of fluorescence, particularly on the cell membrane for this one sitting on top. Now what we also do, because in confocal, we can do z-axis sectioning. So we do sectioning to take two-dimensional image at 0 micron from the surface, which means it's on the surface, and 2 microns, 5 microns. Sorry, 5 micron from the surface, 10 micron, and 15 microns. You see that we can see the fluorescence very clearly, enhancement, not only on the surface, but also well above the surface. This is well beyond the so-called near field enhancement distance. Okay, distance. We also did the other dye to label the calcium inside the cells. Now we're not labeling the cell membrane by labeling cytosol. We are actually labeling the calcium in the cytosol. And then also label the nuclei by Hertz dye. We see a very similar effect that when the cell is sitting inside these small squares, the cytosol fluorescence becomes much brighter. But we don't see a lot of difference in terms of their nuclei fluorescence intensity, because that's actually trigger us to think about these mechanisms that we think most of the energy, light energy, is concentrated or enhanced around the cell membrane. So you don't have a lot of enhancement inside, in the center of the cells.
So what's the calcium do in the biochemical signal by interacting with the molecules differently, or does it have a physical effect?
So
That something is calcium.
Oh yeah. So this is a dye that. Let's see if this. Okay. This is a dye that I may take that back. This is probably just a calcium AM dye that label the living cells. If the cells are alive, you probably see the fluorescence in green. Yeah. So in this case, that we actually show quantitatively, we have a highest enhancement on the membrane. So we do a quantitative analysis about the fluorescent intensity on the cells at different Z locations. We see that the actually fluorescent enhancements happening not only on the surface, but also well above the surface. And the maximum enhancement with respect to the silver surface, to the smooth silver surface, is about 70 times. And if you calculate the actual enhancement time factors with respect to the glass slide or petri dish, that's over 100 already. All right, so we also did some control experiments to exclude the factor that the cell may just behave differently when they are on the nanostructures, okay? Which is my second topic I will touch upon very quickly. But in our control experiment, this is on silicon-based, nanostructures coated with the silver. You can see on silicon, you still have some nanostructure here. We don't see any enhancement. So the enhancement does come from the metal. If you don't coat metals, you don't have the enhancement. On the glass, the fluorescent intensity is even weaker because silicon is a little more reflective than glass. Okay. And we also tried to do is we actually it's not entirely due to. Yeah. So this is showing that without metal layers, there's almost no difference between the intensity of the cell inside and outside. So we did some control experiment to prove that we did have very significant enhancement when the cell is sitting on top of nanostructures for both membrane and cytosol, but more on the membrane. All right, so of course that's just a very rough demonstration. We can enhance fluorescence. But what eventually we want to do is, if we transfect the cell with GFP, or in this case RFP, red fluorescent protein fusion paxillin, which is skeletal proteins on the cells, can we really see that protein expression as early as possible, earlier than most people do? So that's what I get to know from my collaboration. Well, this is a collaboration between me and Peter Wang's lab. They have a very established technique to transfect cell with different fluorescent proteins. And so in their typical case, they do imaging 36 or 54 hours after transfection, because before that, they cannot see a very reliable signals because the number of molecules is too low. They cannot have the sensitivity to see them. But in this study, when we culture the cells in nanostructures, and then we do transfection, and we start doing this RFP imaging only 12 hours after transfection. So this is a very huge improvement over the status quo. Okay, you can see clearly on the top row is the cell imaging on the glass slide or on the petri dish, which is their conventional way, and the second row or the bottom row is the imaging on our nanostructures. You can see a huge difference. So we think this will help biologists, especially a person like Peter Wang, to visualize the protein expression or protein distribution, especially what he's interesting to see how this paxillin distributed on those anchor points along the cytoskeleton areas. Okay, so he cared about that. But what I can provide him is to let him enable the early observation of that, much earlier than before. Okay. All right. So what we are also trying to do is to see if we can do a fast dynamic calcium imaging or enhanced fluorescent lifetime imaging. Because there's something I forgot to mention is when we do imaging, we use a system in IGB. We use a confocal system in IGB. Okay. We use the shortest possible integration time for each pixel. Okay. That also brings us another benefit besides of sensitivity boost, which means that we can reduce the photo damage to the lowest levels because we can let the photon resides in one pixel at the shortest time. Okay. And that means that even when you have high expression level of protein, you can always using our substrate to reduce your integration time on each pixels and to improve your scanning rate. You can basically do much faster scanning, confocal imaging that will allow us to do a fast dynamic calcium imaging or other dynamic imagings. Okay. All right. So, go back to the slides. We actually did a lot of cell growth study on nanostructures, especially on this silicon nanopillar structures. So that actually triggers us to think, is there any nanomechanical interaction between the cells and the nanopillar, especially when the nanopillar becomes very, very small, very high aspect ratio, and very, very flexible. So what we do now is we're trying to create this nanopillar, high-density nanopillar polymer. So you can see a lot of high aspect ratio polymer nanopillar, which are really flexible, really flexible. We can also make those polymer nanopillar on top of a polymer microstructure. This is a microscale pyramid structure we made out of polymer, and we can make those nanopillar on the surface. Then we use the surface as a nanomechanical interaction tools to study the cell mechanics. What we actually observe is very interesting. We try to culture the cell. This is a CHO cell again. Culture cells on this microscale pyramid structure. We didn't see anything special. But when we started to culture this CHO cells on this nanopillar array substrate, we see the cell behave very differently compared to their normal behavior. So let me show you the next slides. You normally don't see the cell looks like this. Some cells really start to stretching them apart to a very, very thin sheet, and the actual size of the cell becomes sometimes over 100 micron. They become crazily big, crazily stretching out. And if you look closely, they actually form a lot of anchor points.
Are they still alive?
Yes, they're actually still alive. They're still alive. Otherwise, they would not spread. They will just sit, on top. So this is more a picture showing that we do have a lot of cells that like to stretch them into a very either very long shape or very spread out thin sheet. That means there's something's going on. There's something underneath it to provide a mechanical cue for the cell to spread itself. There must be something that the cell can hang on to stretch itself. That's exactly the case. We actually observed that many cells from this, a lot of philopodia or tentacle, you can say, they're trying to reach very far away to grab onto those flexible polymer post and use that post as the mechanical cue to feedback the cell growth and migration patterns. You can see that we can also use this tool combined with the electron microscope to study the cell fourth. Because we know what's the dimension of each pillar, we know the material Young's modulus, and we can visualize the bending conditions of each pillar. So we can basically calculate what's the force of the cells exert on each individual pillar. You can see they actually really start reaching out to grow a lot of hands to grab onto those pillars. Yes.
So if you
Okay.
go back to the slides, it seems like the, so, one more. Yeah, so we can see the microarray in the
Yes.
but
Right.
the other part seems like there's no microarray. So
Right. So we actually intentionally create a multiple topology or geometry or device on the same device so we can culture them together to compare them more fairly. In this case, what you see here is a nano on micro stuff. What you see here is this structures. You have nano pillar on top of a micro pyramid. Okay? And the rest of the area just nanopillar itself. All right, so this is the work actually. This is a recent result Xintang just sent me. It is a very, very exciting result. I have a look at it. This is a collaboration between my group and Professor Tahir Saif's group, and graduate student Xintang did most of the culturing work on the surface. So we're trying to compare, can we use this surface also as a sensor to tell whether you have a malignant or benign cancer cells? Okay, here we use HCT8 and use two subtypes of the cells. This is a colon cancer cells. We use two subtypes. One subtype is our subtype, which is much more malignant, much more aggressive than the other type, which is HCT-8E. So we see a very apparent difference between these two. We can see that, especially for these malignant colon cancer cells, it starts to really reach out very long filopodia or long tentacles. However, for this less aggressive cells, colon cancer cells, it has much shorter or much fewer filopodia extended out. That's one thing. Another thing is you can see that most of these malignant cells, they tend to actually stay on the surface without spreading too much. However, for this benign cell lines, they tend to cluster together, also spread out.
Any clue why this?
Well, this is a very new piece of data Xintang sent me yesterday. We're, of course, start to investigate the molecular mechanism of it. That's why we have to combine this together with the molecular imaging to see the actual expression level of our interest proteins on the surface. Okay. But here is another example that if we culture cells on this nano on micro structures, you can see also you have those malignant cells tend to stay as a spheroidal cell without spreading itself, and you have the this benign cell lines trying to cluster each other. All right, so with that, I will conclude my talk, and I want to thank all the collaborators. I'm sorry I didn't put Xintang's name here, but I want to verbally acknowledge him and also acknowledge our collaborator in Lawrence Livermore National Lab to help us, in some cases, help us to do the large-scale device fabrications. And I have funding from various sources. I want to thank them too. Okay, thank you so much.
So it's great to see such a wonderful technology in our immediate neighborhood. So we'll see what we might be able to use it for. Maybe some ideas come spring up now.
Yeah. Okay, Paul.
There's still the question about how much the photostability goes up, how much the excited state lifetime goes up, and apparently there's some disagreement.
Yes, right. So we actually calculated all the stability, quantum yield based on those equations. Of course, those equations could be wrong. Could be, or the actual coefficient could be different than what we actually predict. If you go to paper, there's a paper in APR. Well, this is the data we measured experimentally. And if we trust this data, and then we use this data to then calculate our radiative and non-radiative decay rate, gamma, and then use that to calculate our quantum efficiency Q. Okay. So here we actually see both change of the radiative decay rate as well as the non-radiative decay rate in our case, based on our measurement.
So what are they? How much does the lifetime go go down, and how much does the quantum yield go up?
Well, I didn't have the table here, but it actually varies from molecule to molecule. It's really molecule dependent. Okay, the highest one, I think, for the quantum efficiency wise, we can improve the quantum efficiency. The best case we have is about 4 to 5 times quantum efficiency enhancement. For some high quantum efficiency a fluorescent molecule like Rhodium-6G will only have very little change.
Into the space.
Yeah.
How far?
How far? Okay. For this study, we only study the molecule absorbed on the surface. We didn't.
In principle, does it have to be within
Yes.
nanometer?
Yes, they have to be within 100 nanometers above the surface. Yeah.
So you see only really the interaction with the membrane of the cell.
Yeah, so we think this is due to more than just electrochemical electromagnetic enhancement. This is also due to the non-radiative near-field energy transfer between the fluorescence molecules and the plasma substrate.
No, when we put the cells on your surface, they do new things which we might not intend. But let's say we say, oh, this is interesting, let's study it. What is your understanding why they, for example, step out or draw up?
Yeah, we have,
Is it a patterning,
Yeah.
or is it a mechanical rigidity?
We have a very little understanding right now. That's why we have to do very systematic studies to vary the material property, to vary the shape of the material, of the actual structure, the size, as well as to change the cell types. Because we noticed that for different cell types, they behave differently. Okay. For example, this is a case that we cultured the CHO cells on a silicon nanopillar device, something like, well, you can imagine in your head that we have a lot of silicon nanopillars inside the squares, and the cells looks like okay to us because the cell does not exhibit a very dramatic philopodia to us, or the cell didn't spread crazily to us. But for the same cell, if we do this on a flexible polymer nanopillar, the cell becomes really different. So this is cell culture on this flexible polymer nanopillar substrate, and that started showing this very, very interesting philopodia effect. So there's a lot of there are a lot of factors involved here, and we just start to investigate this. So we're going to do a very systematic study.
Do you have any clue what leads to the difference?
Well, it could be, we have a lot of hypotheses. So the actual clue here is, the reason why for the cell behaves so differently because this flexible nanopillar really provides a nanomechanical feedback to the cell. The cell has a force sensor itself.
So the pillars are themselves wiggling.
Yeah, right.
Okay. And the cell interact with the integrins or?
That's about that we have to do the molecular imaging to visualize those cytoskeleton proteins to see whether
the extracellular matrix proteins.
Exactly.
The nectins.
Exactly. There's a lot of work for us to do. Right.
Okay.
Yeah.
Isn't cell differentiation for another type of cell, something like that, or no? Like if we're trying to put some stem cells, could it be inducing some?
Could be. It could be, but we never tried stem cells so far.
This kind of cell, it could happen, no?
In this kind of cell, the cell may, yes, we have to do property sequencing to see whether cells really develop or differentiate into different subtypes with the feedback of this nanostructure. Yeah.
If you put the bacterial cell on there, they're probably pretty inert, right? Because they are rigid and they always just lie there and don't do much. So it's more for eukaryotic animal cells or this kind.
Yeah, we haven't tried. Well, we do have a project based on E. coli and Salmonella and yeast, but we never tried to culture those kind of cells on this substrate yet. We want to focus on, the actual methodology for engineering department may be slightly different from physics department. We want to find a solution or provide a tool first before we dive completely into discover this physical mechanism. There are two, actually, this is very, very good. We have to know everything, but that actually takes a lot of years and also a lot of studies.
An engineer that works, that's fine, even if you don't know why it works.
Well, of course, to actually make it work better, you have to understand it better. But what we try is, I want to do this in parallel. Simultaneously, we want to find the engineering application of it. When we see a very interesting observation, we want to find the actual biological applications. In the meantime, we also delve into physics, biophysics, trying to understand why this is happening. Yeah. So for example, in this case, even though we don't understand that quite well yet, but we think we maybe need to use this plus the optical microscopy or electron microscopy to do a differentiation between different subtypes in cancer cells.
Yes, questions?
Pillar? So all these results, you have a silver everywhere. Silver is everywhere, inside or outside. That's why we need to use pillar. Without pillar, you don't have this high enhancement.
Can I ask a question?
Because the field is more stronger near the pillar tips, or why?
The field is, in terms of localized field enhancement, yes, the field will be concentrated on there, on the tip of the pillars. But here we're also talking about, you have some surface plasma wave going on this film, and also you have multi-scattering going on the film. And those are radiative processes, and they could be coupled inside the cell. So that's our hypothesis here. We do have localization of EM fields somewhere here. You can see some hotspot right here, but there are also some part of energies coupled inside this cavity, spherical cavity.
Yeah, a very quick question. So, what is the time resolution for this technique? For example, like it's a very large part, when we see the stretching out the cell, can you get to like some time domain information, like?
Yeah, sure, absolutely. So I only show you a very little bit of result here, but we do have a 24-hour, 48-hour culture video to show how the cell behave differently, how the cells start to divide, to spread, to migrate. Yeah, we do have that. And we're doing very systematic analysis of the video. As you see, as you can see, imagine the actual video file is big and also have a lot of cells in that video. So we are doing the actual analysis of each individual cells by tracking them down for 24, 48 hours. Okay.
Your bright field images of the Chinese cancer cells?
Yeah. Here. This is bright field.
It looks to me like there's actually a pretty high fraction of those that are spread out even when they're not on this microfabricated surface. So when you claim that when they bind to this, these pillars, it makes them spread out more, have you quantified that? Is that
Yes, we haven't. I'm sorry I didn't show the SEM picture showing for the cell stretching or cell spreading on the smooth silver surface. We do have the comparison. And of course, you can see some cell, maybe a small number of cells stretching really long. But when we culture this on the nanopillar surface, you have large number of cells stretching to over 100 micron long. So there's a statistics here that we actually already did, and sorry I didn't show the SEM here, and I should have shown to do a side-by-side comparison of that. Okay, good question.
So when you start talking about trying to reach low copy numbers, it seems like coupling to microcavities would be problematic because you're going to be biasing yourself toward imaging things near the membrane. Can you comment on if you think that's going to be a problem for sort of regular living cells, which wouldn't be spherical, but
yeah. Right. So here, right, we are I mean, I already said this. These hypotheses only apply to those spheroidal cells, especially when a cell looks like a sphere. It actually works very well there. And when the cell spread it out, I think most of the enhancement coming from the surface. Because when the cell is spread out really thin, the actual thickness is only a couple of micron over there. So you can take advantage of a near field enhancement, pretty much in that case. Yeah, so for the cells, like, especially for cancer cell, it likes to grow into this three-dimensional spheroid, like in this colon cancer case. You can also find that very similar behavior for breast cancer. It tends to grow into a spheroid. So in that case, we think this technique may help more to enhance the actual sensitivity in our imaging.
Okay. Okay. We have one more question.
Okay.
So the cells you show on the yesterday image, those cells are dead, right?
The cells are alive before we fix it.
Okay.
We actually fix the cells, then we do staining, and then we do SEM of the cell.
My question is, during the cell from live to death, do they change the morphology, changing from spreading on?
We don't think so, unless you don't trust the so-called fixing process. Because a lot of people do fixing cells. Of course, there might be something happening during the fixing process, but we think in terms of morphology, because it's actually a fixing process. I'm not sure have you done that before. It's very quick process. So we don't think within that short time, the cell morphology will change that large. But of course, the actual best way still, we have to analyze our video microscopy to do imaging.
Can we guess in terms of time? Can you give is it a second? Is it a millisecond?
Well, it definitely depends on whether the question is skillful or not. But it's only, I think you can finish the work, finish the actual work in tens of minutes. So we don't think the cell morphology will change that large within that short time. Yeah.
Okay, good. So let's thank the speaker once again.
Okay.